one of the reasons we've landed on focusing on opioid use disorder, beyond that it's obviously a critical public health crisis, but it's also just an incredibly complex trait. We're talking about genetic risk, genetic factors being involved, as well as environmental, lifestyle. It's a disease that will only happen if you're exposed to the drug itself. And so that kind of interplay between genes and the environment, I think, is the next frontier of our understanding of genetics in general. One of the sort of standard ways to understand genetics of disease, and which gives us an insight into how these diseases work, is to collect really large sample sizes. So get a lot of people who are afflicted with the same disease and a lot of controls. We're talking about hundreds of thousands of patients being enrolled into a study, looking at their genetic information and looking for differences. And this strategy has been really successful for a lot of different diseases, but opioid use disorder is sort of behind the curve. So a lot of the interesting things we've learned from genetic studies over the last decade has been the role of enhancers. And so this is an incredibly important part of our biology. It allows your heart cells to contract and your neurons to send signals, is by controlling which genes are turned on and off. And so we wanted to directly measure gene regulation effects in these opioid cohorts. So aiming to identify what types of enhancers and regulatory elements are different in patients who are afflicted with opioid use disorder compared to healthy controls. Yeah, so a standard genetic study will be looking for hundreds of thousands of patients to pull out which genetic changes are specific to our patient population. What we're doing is a much smaller cohort. We just looked at 100 different tissue samples, 50 people who died of an opioid overdose and 50 controls. And even in this small cohort, because we are directly measuring what's happened in the brain tissue, we were able to pull out some really interesting differences. What was most fascinating to us is that what was different was not necessarily consistent in every patient that we saw, but where we saw the differences was. So even though these little changes in gene regulatory elements were distinct for each person, they each had their own unique change, they happened around the same set of genes. We now have a spot in the genome to focus on. So we found these genes that we think are really different in the case of opioid overdose. But our next step is in figuring out what part of this disorder are they involved in. Is it what makes you more prone to a lower pain tolerance, and therefore you require a larger dosage of opioids when you first have an acute injury? Could be something that happened earlier in your life that altered the way your cells function. So teasing apart those differences in causation is how we're going to get the next step of insights into the disease. We're currently working on a variety of strategies to investigate these genes, mostly looking first at what they do within the cells themselves. So how does this intrinsically change the way neurons work? Certainly we know there are certain functions that are prescribed to different regions of our brain, like the occipital lobe helping us see, these sort of connections. But whether those differences are done through neurons communicating with each other or because the neurons themselves are different is unclear. So we first started by collecting samples from different brain regions and using a technique that allows us to look at which enhancer or regulatory elements are functional. And in this work, we're starting to pull apart some enhancers that are only active in certain brain regions. Most excitingly, the enhancers that are specific to a region of the brain called the nucleus accumbens seem to have a connection with substance use disorders. And the nucleus accumbens is the part of the brain sort of classically linked to your reward circuitry, so how you respond to a positive stimulus. Why do we care about why the disease works? It's because that's how we prevent it. If we could just understand who was at risk for this disease, we could think about ways of increasing monitoring, changing dosage, giving alternative pain management strategies to people who are at really high risk. We have to be really careful here because these are really critical public health questions, but if we could use what we know to help people understand their risk level, then we might be able to give them some really practical information to help them protect themselves.